represses transcription. Co-repressors help them, but don't bind in a sequence-specific manner. But these co-repressors, and here's some examples, recognize the methylated DNA, and these, for example, are histone deacetylases. Histone deacetylases remove acetylation of histones. Acetylation histones is a positive mark. If you remove the positive mark, you're more likely to be repressed. So what happens in cancer is that these promoters that usually aren't particularly methylated become methylated. How that happens, we don't know. But the good news is that these changes, like DNA methylation, they're made by enzymes. They're put on by enzymes, they're removed by enzymes. So the thing about enzymes is you can inhibit them. And so there are inhibitors of DNA methylation. This is just one example, that when you give it to cancer patients or tissue culture cells, the result is you now lose that methylation and you can turn tumor suppressor genes back on. So there's a lot of excitement about these kind of drugs, we call them epigenetic drugs, for reversing these changes to gene expression that occur in cancer. And so the great thing about these epigenetic changes, which are themselves regulating gene expression, you can reverse them. Now, genetic changes that occur in cancer are permanent. You cannot reverse a DNA mutation. Not yet, anyway. Maybe one day. Maybe one of you in this room will develop the technology. Sure.
So, does hypomethylation lead to any disease states?
Absolutely.
So, would there be
hypomethylation?
Yes.
There are many mutations in the enzymes that either methylate or that bind to the methylation. So then, yeah, that would be hypomethylation. That lead to genetic diseases, Angelman syndrome. There's a bunch of them. I don't have a list. They're all neural diseases. People have only very recently found the enzymes that demethylate DNA, and so those will now be targets of developing drugs. Rett syndrome. There's a lot of imprinting disorders. It's actually interesting when you do in vitro fertilization, because the eggs are grown outside the body, you lose methylation during that process. And so, I mean, I know this because I got triplets through the process, but there's a much higher tendency for those kids that are from in vitro fertilization to have these diseases of methylation defects. There's a whole bunch of names that I don't remember. I'll email you. Prader-Willi syndrome. But yeah, it's definitely a part of the puzzle that if you change the methylation leads to diseases. Okay, so in general, a lot of the methylation in a healthy cell is in heterochromatin, DNA methylation, and CpG islands are under-methylated, but during cancer, this kind of switches. We lose methylation in these heterochromatin regions, and this actually leads to genomic instability, and we gain methylation in CpG islands that leads to repression of tumor suppressor genes and other genes. Okay, so methylation is something that can be, it is inherited through cell divisions. And that's because both strands of the DNA are methylated. So it's a CpG, right? So on the other strand, you've got a CpG. So you've got a methyl on one C, a methyl on the other C, on the other strand. I guess that's why they're in CGs. When DNA gets replicated, the DNA is transiently hemimethylated, but then there are maintenance DNA methyltransferases that come in, recognize the hemimethylation, and put that methylation back on the other strand. And so this is a histone, well, it's not histone, it's a chromatin mark that is unarguably inherited. Whether histone modifications are inherited or not is a big question. I personally don't think they are, but it comes back to why people in chromatin say they're doing epigenetics. It's just an infighting that you're going to hear more about. Histone modifications are not epigenetics. I'm sorry. DNA methylation is epigenetics, meaning it can be inherited through cell divisions. Okay, so as well as this maintenance of methylation during replication, you can also get de novo methylation. So when you want to change your euchromatin to constitutive heterochromatin, you get other types of DNA methylases coming in, making new methylation. And very recently, several mechanisms have been discovered to actively remove methylation. And this is something that has to occur during, soon after fertilization. And just for the sake of time, I think we'll leave imprinting. Oh, here we go. I do have a list. Just for you, here are a list of diseases that are due to mutations in either the DNA methylases or methyl binding proteins. And these are inherited, and these, I guess, are spontaneous. So detection of methylation. I guess the only thing I want to tell you about this is there are different ways to detect methylation, but by far the most used method is what's shown here. And that is when you treat, so if you sequence methylated DNA, it's going to be a C, whether it's methylated or not, from the sequencing machine. But if you treat your DNA with bisulfate, it deaminates it. Basically, it removes. All right, give me a minute. If you treat methylcytosine, so it's got a methyl here. It doesn't look like a uracil. When you bisulfate deaminates. If you treat cytosine with bisulfate, it removes the amine and it now looks like uracil. So uracil base pairs with what? Adenine, right? It's the RNA version of T.
Yeah.
So basically, you treat your DNA with bisulfites, sodium bisulfites, and it changes your unmethylated cytosines to a uracil, but not your methylated cytosines. And then during the process of sequencing, the sequencing reaction is going to put an A opposite the T, whereas it will put a G opposite a methyl cytosine. So just a simple treatment of your DNA sample with this chemical can allow the methylation to be revealed in sequencing. And so this is something that is very broadly used now in patient samples and otherwise. So this is just another example. Oh, okay, so this might help a bit better. The other slide was confusing even to me. So the unmethylated Cs, when you treat with bisulfate, are going to become Us, and the methylated C will stay as a C, and then you sequence it. Obviously, you have to know what the sequence of that DNA needs to be before you start. You can't do it with, some strange organism whose sequence you don't know. And these are the things we just kind of filler. So the main things about epigenetic changes is that you can reverse epigenetic changes. Another example is with the use of histone deacetylase inhibitors. And again, these things don't occur in isolation. They're all working together in a concerted manner. That is the post-translational modifications of histone tails, moving nucleosomes by ATP-dependent remodelers, and adding extra proteins like the histone variants or proteins like HP1 that lock the chromatin into a more open or a more closed down structure. And so basically what the chromatin allows the cell to do is to regulate the processes that occur on the genome by changing access to the DNA sequence. It allows the cell to respond to external stimuli. I mean, often signaling molecules, their final function is to modify a histone to influence chromatin structure, and at least DNA methylation is epigenetically inherited. And so I love this picture. I don't know who thought of chromatin in a manner like this, but just to tell you that during metaphase, the chromatin is extremely highly packaged, where whatever level of packaging that exists in interphase, I've never heard that word used, so never bothered to learn that in my 20 years in the chromatin field. This is just a strange slide altogether. Sorry. Even the color choice is interesting. I don't know, but so during metaphase, you do get this really high degree of folding to this chunky chromosome, which functions to shorten the length of the chromosome so that when you pull them apart, they're not ripped apart. And this is just a model, much like the animation, for how this chunky, highly condensed chromosome might form. We have no idea if this is true or not because visually we can't see if that's the case. So the centromere, that's where that constitutive heterochromatin is, that packages it very tightly. Telomeres being the end of your chromosomes. And again, just to show you that this histone variant, SEMP-A, when you do immunofluorescence, you see it's right on the outside edge of the centromeric heterochromatin here, where it attaches to this huge mass of proteins called the kinetochore. And this is really important for making sure that this chromosome attaches to one spindle coming from one pole, and this attaches to a spindle coming from the other pole, in order to accurately separate those two chromosomes, one into each cell. And this is just really by way of background for the lectures that Jill Schumacher will tell you on the roles of a lot of mitotic kinases. Okay, so we are done, and anyone has any questions, please come and ask me.